Now, this is a topic that I'm personally very interested in, both from the perspective of a health coach that works with genetics and nutrigenomics, as well as a long-term cannabis user. And my relationship with cannabis has really changed over the years, as I imagine yours might have too. And as I began to look into some of this research on how genetic variables affect your response to cannabis, a lot of my experiences I've had with friends and other people really started to make a lot more sense. For me, I remember discovering pot when I was about 14 or 15 and literally just not understanding that other people didn't get the same kind of positive effect out of it. And, the first time I smelled it, it was like the gates of heaven had opened and I needed to know what it was. And looking back, it was a real innate biological response. And we'll see later in the webinar that your genetics really influence this type of response mechanism, the dopamine reward centers in your brain, for some people a lot more than others. I was the kid in high school that tried to get everyone to smoke with me because it really normalized me in ways that I'd never experienced before. But as I've gotten older, I've realized that it makes me a lot less functional in certain ways: memory, attention, motivation. So I tend to move back and forth between using it and not using it. And again, looking at this from a genetic perspective makes a lot of sense as well, because a lot of the genes we're going to cover here have to do with memory and cognitive function. And you'll see that there's really a pretty major difference in how people's brains respond on a cognitive level. Looking at our relationship with cannabis from this genetic perspective is going to be really helpful in unraveling why it's such a potent attractor for some people and why some people could really care less. And I think taking this kind of personalized individual perspective is really necessary to get a clear picture. There tends to be a lot of ideology involved in cannabis. People tend to be either anti-cannabis or pro-cannabis. And, let me be clear, from a legal standpoint, I'm for legalization, especially for medical patients. But looking at cannabis through this lens of genetics and individual metabolism kind of cuts through this false dichotomy of either marijuana is the devil's weed, or if everyone smoked it, the world would be a better place. I don't think either of those are true now. And this webinar is really designed to kind of cut through the rhetoric and look at the science that's out there. And as we move into cannabis and other cannabinoids being used more readily for medical purposes, it's going to be really important to have this knowledge in our tool belts as practitioners, so we can make informed decisions for our clients. And also, just in general, as casual users, you can really start to understand yourself a little bit better. And I think we're going to see some really remarkable things come in the future around personalizing strain selection and breeding with this type of genetic information for people. And just to get this out there, I want to say I'm not telling anyone to smoke cannabis or not to smoke cannabis with this webinar. That's for you to decide and for you to understand the legality of it where you live. But before we go into the genetics, I just want to quickly look at some of the conflicting science that's out there on cannabis, which can make it very confusing. And we'll see as we move forward, some of this conflicting information is in these studies is really a result of not looking at these genetic variables as a factor. So let's just go ahead and look at some of this data here. So I just pulled a couple excerpts from some summaries, some abstracts from some articles, some studies, and we'll just read these couple sentences and you'll see how varied these results were. Very heavy use of marijuana is associated with persistent decrements in neurocognitive performance even after 28 days of abstinence. So that's from one study. Another study, more intriguing, it appears that the concurrent use of cannabis has no compounding effects on cognition, as well as emotion/affect-based decision-making. Here's two more. Some cognitive deficits appear detectable at least 7 days after heavy cannabis use, but appear reversible and related to recent cannabis exposure rather than irreversible. And another one, more frequent cannabis use was associated with better performance in attention and working memory tasks. So right there, you see there's 4 completely different conclusions. What's the deal? The tendency generally I find is people want to point at bias from, people who designed the study, perhaps those people were anti-cannabis or pro-cannabis to begin with and were just looking for results. Maybe there was small sample size, which could have skewed the results, or maybe there was poor study design. But I'm going to make the argument as we move through this, especially related to these are studies are on memory. I'm going to make the argument that because they weren't looking at genetic variables, they really couldn't get a clear picture on what was going on. And the studies we're going to look at later really hammer that home, I think. So I just want to give you a quick outline of what we're going to talk about. I'm going to spend about 15-20 minutes just kind of going over some basic genetics. So you can really get a platform and basis for understanding what I'm talking about when we start talking about genes and cannabis. I don't want to spend too long on it, but I just want to give you a background. And then in part 2, we're going to talk about 4 different genes that have pretty solid data behind them. And we'll mention a couple other genes as well. And then I'll take some questions. Okay. So DNA. DNA carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms. I think we all learned that in high school biology class, but how is that? That is really amazing, and it's so deep. But at a basic level, it's made of these 2 strands of nucleotides, which are the repeating code of DNA, A, T, C, and G. And the specific pattern of these nucleotides goes into creating proteins, which go into creating your traits. And we'll kind of walk through that process just a little here. So there's about 3 billion base pairs in your DNA, and 99% of it is the same as every other human's DNA. So the 1% difference is what makes my hair brown and curly, maybe makes your hair blonde, and it's what causes these differences in the way we metabolize different foods and substances. Everything from saturated fat to carbohydrates to different herbs, and as you'll see, cannabis. So here's a nice illustration of how these 2 nucleotides pair together. Those are called base pairs, and which letters they are going to be what's called an allele. And I'll use the word allele a number of times throughout the webinar. So just know that that's referring to which letters are in the code at a specific place. This whole process here is called the central dogma of biology, where DNA has this code, RNA comes along and reads the DNA, and actually I'm going to go ahead and make my slides a little bit bigger. I'm going to minimize myself here. I think that'll help you guys see this a little better. Just one moment. Yeah, there we go. And then RNA comes and brings those instructions over to what are called ribosomes, and they make proteins. They assemble proteins using the instructions. There's another picture of that. You can see the DNA, the RNA comes and brings the instructions over to the ribosome, and then that creates all these proteins that go into creating your biochemistry. And if you want to think about it as a metaphor, the DNA is kind of like a recipe, and the RNA is a little bit like a chef, and the ribosome is kind of the final assembly of the ingredients. And we'll kind of come back to that metaphor later on here. So what's a SNP? A SNP stands for single nucleotide polymorphism, and it's essentially a 1-letter difference in that code from person to person. They're fairly common. They occur roughly every 300 base pairs. Every 300 letters, you'll get around a SNP. And there's over 10 million SNPs in human DNA. So this is a very common thing. This isn't something that's rare. SNPs are what make you, you, and what make me. And as scientists have begun to really decode, what these SNPs do, we're really getting a better picture on individual metabolism. It's really revolutionizing the health industry. And we're going to see a lot more of this come online, next 5-10 years. It's going to be a lot more commonplace for doctors and other health practitioners and coaches to be working with this. So what do SNPs do? They create the original recipe for your unique biochemistry, and they influence the way you respond to the environment. They influence the way you respond to different foods, different toxins, the way you respond to exercise. They kind of really shape who you are on the chemical level. And they can be used to assess predispositions, likelihood, disease risks, that kind of thing. They can help you understand your individual metabolism, and they can be used to create personalized health regimens. And that's what I do as a coach. I look at people's SNPs and help them kind of narrow down what the choices are and really give them a better idea of, what's going to work well for their unique biochemistry and their body. And SNPs are most useful when viewed from a systems perspective. Hey, Mark, no problem. Glad you're here. SNPs are really most useful when they're viewed from a systems perspective. And I'll come back to this a couple times, but you can look at one SNP and get a picture, but when you really start stacking them together and see how they interact, that's where the magic is. So epigenetics, I'm not really going to talk too much about that. I'm not really going to talk about it at all. I just want to mention it because it is a factor, and it's one of the things that kind of creates more of a probability factor than a hard line with these SNPs. Because what epigenetics is, it's essentially the control over genetic expression. So genes can either be turned up or turned down. So as they produce those proteins, there's certain factors that can come along and influence them to produce more of those proteins or less. And I'll just show you one little possible epigenetic modification that's really neat. When I first learned about this, it kind of blew my mind. But we usually imagine DNA as that double helix structure, but in order to fit in the nucleus of a cell, it has to be wrapped very, very tightly. So it ends up being wrapped around these proteins called histones. And what happens is an epigenetic mark, either an acetyl group or methyl group, and you don't have to know any of this, it's just really cool information, will come and influence how tightly that DNA is wrapped around the histone. You can see here how loose it is versus here how tightly wrapped around it is. And what that does is it either allows the RNA to read that DNA and turn it into a protein, or if it's too tightly wrapped, the RNA can't actually get in there and read the DNA. So you can see just from this one particular thing that, you might produce more or less of a protein depending on how you respond. And epigenetics are really in response to the environment, really in response to different stimulus. You get these changes over time. But that's just to keep that in your awareness. We're not going to talk too much about that. Are SNPs mutations? No, they are not. And it's a common misconception. People often call them mutations, but it's only considered a true mutation when it's in less than 1% of the population. SNPs are really naturally occurring variances. And let me go back a couple slides here. I think I'm mispointing something out. Yeah, so this diagram really illustrates what a SNP is nicely. So you see in example 1, this person has a C right here. And then in example 2, this person has a T. And at the very basic level, that's what a SNP is. It's just one letter substitution. So I want to give you 2 example SNPs that don't have anything to do with cannabis, just so you can kind of see how they work in the body. And then we'll talk about cannabis. So this is a SNP called CYP1A2. It codes for a detoxification enzyme in the liver. So it creates a protein that acts as an enzyme that breaks down certain substances, and it's responsible for breaking down caffeine. This number right here, the RS number, is the identification number. And as we go through this, each gene that we talk about, each SNP, is going to have a particular RS number. And you can go use those on your own if you want to look up, if you have your 23andMe done or another gene analysis service, you can go look up your SNPs using these RS numbers. So variations in this gene at this particular SNP, 762551, are associated with either having a slow metabolism or rapid metabolism of caffeine. Now, the AA allele is a rapid metabolizer, and anyone with a C allele, either AC or CC, is considered a slow metabolizer. And what this really means is that the slow metabolizers just don't produce enough of the CYP1A2 enzyme when they're exposed to caffeine. So they don't break it down fully. Whereas the AAs break it down fully and seem to be able to drink multiple cups of coffee and continue to break it down. What does this mean? This is a chart taken from a study. I believe this was done in 2006. And what it's showing is the risk of hypertension and heart disease associated with how much caffeine someone drinks relative to their CYP1A2 gene. So you can see, and this vertical axis is the odds ratio of getting heart disease. And the AA genotype here, you can see as they drink more coffee, their risk actually goes down. So these are the people that completely break down caffeine. And the CC and the AC here are the exact opposite. And this is a really nice linear kind of response. But as they drink more coffee, up to 4 cups, they can quadruple their risk of getting heart disease. So this is something that, is really important to know, especially if you're a coffee drinker. Myself personally, I'm a slow metabolizer, and I switched to decaf after I learned this, which kind of kills me as a coffee snob. But I think it's the smart thing to do in the long run. And, this is something that, it's pretty important to know in terms of looking at your long-term health. And one thing, if you don't know and you don't care to get your 23andMe done, note that drinking 1 cup a day is going to carry the exact same risk. That's totally the same, but it's just when you start to drink more coffee that you have problems with these slow metabolizers. So ApoE, this one is a really big gene. There's a lot of research on this. And this codes for a protein that influences cholesterol and triglyceride balance, as well as arterial plaque formation, the hardening of arteries. It also influences brain inflammation. And it's actually determined by a combination of 2 different SNPs in the same gene. And so they classify this either as a 2, 3, or 4, and you kind of add both of those together. So you can be a 2-4, a 3-3, a 4-4, etc. But what they found is this ApoE4 variant, which is 25% of the population, carries a much higher risk of dementia and Alzheimer's disease. And what's even more interesting and really important to know is that this isn't just something that's like a diagnosis. This is something that you can really modify with your lifestyle. Moderate saturated fat intake doubles the risk of dementia for these ApoE4s. Physical inactivity doubles the risk again. And high saturated fat intake, as opposed to moderate, 7 times the risks of cardiovascular disease and Alzheimer's. So that's a pretty big deal. I'm talking about 25% of the population has the 4. Now, this is a nice chart looking at approximate lifetime risk of Alzheimer's based on the ApoE genotype. Now you can see the 2s and 3s have a fairly low risk, but once you add the 4, or once you add the 4 variant into here, you really see the risk start to triple. And then comparatively to the 2 2s, the 4 4s have about a 6 times risk just in general, regardless. So this is something that's really important to know when you're thinking about what you're going to eat, how are you going to, do your life, and these are just 2 of the genes that I look at when I work with clients. I look at about 300 different genes, and you really start to get a bigger picture when you start to look at a lot of them together. So what are the limitations of SNPs? Well, they're less useful in isolation, like I mentioned. You got to ask, how do they work together as a network? They provide likelihoods, not absolutes. Epigenetic factors can help change some of these things and push things one way or another. And, for example, the ApoE4 variants, not all carriers get Alzheimer's. Why? And you have to look at all these other factors that go into creating kind of the ultimate outcome. These are things, again, like lifestyle, environment, stress, diet. And also trying to figure out SNPs can be very confusing, even with some of the direct-to-consumer reports out there. Someone recently just sent me their report that they had got from a service and was asking me to help them figure it out. It can be confusing to try and sort through these studies just because, it's a lot of scientific jargon. So summary in this part one, SNPs change the function or amount of gene products. So they change the shape of proteins, how they're structured, or the amount of that protein that gets produced. SNPs can be used to help us understand how we work at the chemical level and can give us an idea of predispositions and tendencies. And they can also be used as a guide for lifestyle modifications, such as diet and exercise. And in the next section, we'll see how they can be used to kind of predict how you respond to cannabis and shed some light on why people respond differently. And here's another little chart that just succinctly expresses that. SNP goes into creating the protein, which goes into the cell process, which goes into the trait that you see expressed externally. So let's get into cannabis. What genes impact how we respond to cannabis? Right now, the research is really in its infancy in this, but there is more of it than you might expect. What we know is that THC and CBD metabolism genes affect it. Endocannabinoid genes, what your own cannabinoid system is balanced, how it's balanced goes into affecting how you respond to cannabis, as well as the dopamine reward centers in your brain. So most of the studies so far have approached it from an addiction standpoint and a problematic behavior standpoint. And that's largely because in terms of grant funding, that's what people have been able to get clearance for. And the research climate's been very difficult to study it outside of that. So in general, the research has been very limited compared with other plants and natural substances. I mean, you look at caffeine, there's thousands and thousands of studies on caffeine. It's actually the most studied compound of anything, pretty much. But what I'm going to do is I'm going to try and take a little bit more of a balanced approach and interpret some of this research in a way that can be useful for people that either use it casually, or you might if you're a practitioner, someone who works with clients, you can take this information and start to see patterns in your clients. And as an example, when we get to the CNR1 gene, we'll talk a little bit more about that. That's the CB1 receptor. But I recently had a client who wanted to stop smoking cannabis as much, but really was having trouble with it. And we looked at his genes and found that in general, he had a very underactive cannabinoid system just from the get-go, and was likely trying to balance that out with cannabis, smoked cannabis. But we moved him to CBD, and that really helped him get away from feeling the craving to smoke it, because that was balancing his endocannabinoid system on its own. We'll talk a little bit more about that when we get to that gene. So let's start with COIP2C9. This is pretty similar in a way to the caffeine gene we talked about. It's part of the same family of liver enzymes. And this is responsible for breaking down THC. Now, you can see in this chart, you don't need to know anything about these chemical structures, but THC, when it enters the bloodstream, enters the body, when it comes in contact with COIP2C9, that'll convert it into this 11-OH-THC, which is also psychoactive, and then into THC-COOH, which is an inactive form. And the THC-COOH, when you hear people talking about THC staying in your system for 30 days, it actually doesn't. It actually gets broken down fairly quickly, but the THC-COOH is what stays in your body. And that's also called a metabolite. When something gets broken down in these steps like this and transformed, the COOH form is a metabolite of THC, and I'll use that term a couple times. So COIP2C9, the studies show that it influences how high you get from edibles, and influences how long you stay high from both smoking and edibles. This is the RS number if you want to look this up for yourself, 1057910. And similarly to the caffeine gene, there's a fast metabolizer variant and a slow metabolizer variant. The C allele is about 15% of the population of European descent, and it's relatively uncommon in Asian and African populations. So the AA variant breaks down THC very efficiently, and the CC tends to not break it down. So this is from a study. This is looking at the average amount of THC in people's bloodstream over a particular amount of time. And you can see here that this is about a threefold difference on average between the A's and the C's, or sorry, the AA and the CC. And the AC kind of falls in the middle, but is a little bit closer to the AA. So they're kind of a fast intermediate metabolizer. So what is that? Oh, and sorry, this is another way to visualize it. And this is looking at the specific points in time. So you can see the AA, they immediately started breaking down THC. This is from a study where they're given oral THC. So you can kind of liken this to what it would be like to take an edible. And there's a little bit of a difference with this gene and this enzyme between smoking and eating cannabis, because when you smoke it, it goes into the bloodstream and into the brain much quicker, and CYP2C9 doesn't have time to start working on it. Whereas when you eat it, CYP2C9 is especially present in the GI tract, so it starts breaking it down right away. So that's why there's a difference between smoking and edibles. But you can see with the CC, I mean, their levels are just off the charts, and it takes them, about 72 hours to come back to where the AA variants were at 48 hours. So what does this mean in terms of subjective effects? Well, the CC carriers reported much more intense effects and more drowsiness from the same amount of THC. And this is a particular kind of standardized test, the Stanford Sleepiness Score. And you can see AA compared to CC. The CCs reported way higher drowsiness right away, 2 or 3 times the amount of the AAs. And what's really interesting to me is 3 days later, they reported equal amounts of drowsiness to that kind of initial onset. So if you or someone is someone who just gets laid out from edibles, there's a very, very good chance that they're a CC variant. And this is something to keep in mind, when we talk about medicinal cannabis and dosage. If say for example, someone's never used cannabis before and they're considering using it for a medicinal purpose, this is something as a practitioner to be very, very aware of when you're working with their dosing regimen, because you don't want to just give someone, you don't want to give a CC variant the same dosage you would as an AA, because they might have a negative response because it might be too strong, and vice versa. The AA might need a little bit more than you expect. So as this becomes more available, especially this type of testing, I think this is going to be something that we see used a lot more. And I hope it becomes something that is a lot more, present in everyone's consciousness when it comes to this kind of thing. So CYP2C9 also influences drug tests because, like I said, THC-COOH is the main metabolite that drug tests look for. And CC carriers they barely produce enough THC-COOH to even register on a standard urine test. They produce about 85% less of this metabolite than the AA carriers. And, if you look back at this chart, if you're not converting THC and you have more THC that stays in your system with those slow metabolizers, you just don't have very much of that metabolite, the THC-COOH, in your system. So there's a really excellent website called Professor of Pot. Shout out to the Professor of Pot for really kind of breaking the story on this, that these slow metabolizers, because they don't produce hardly any THC-COOH at all, are more likely to be able to pass a drug test just after a couple days, as opposed to the about 30 days that it would take the AA variants. So this is something also, very important to know about yourself, and probably something that people that are testing should take into account as well. So CYP2C9 summary. And also I wrote an article on this that goes a little bit more in depth. And I'll send out that link after the webinar as well. But AA is the most efficient CYP2C9. You break down THC most fully and rapidly. Likely to experience the least amount of drowsiness from pot, gets less high overall, and their highs shorter in duration. And CC, they have the least efficient CYP2C9 enzyme. They're more sensitive to cannabis in general. And they just don't break down THC as completely. So they're likely to experience the most drowsiness and intense effects, and they get way more high from edibles. And their high, it's just smoke, the high duration is longer as well. So that's the CYP2C9 gene. And I see someone saying that, yep, they're an AA. So the next gene we're going to talk about is something called FAAH, and this codes for something called fatty acid amide hydrolase, and it breaks down anandamide, which is one of your body's two main cannabinoids, the cannabinoid molecules in your body that you produce naturally. And the research that's come out around the endocannabinoid system is just absolutely amazing. I mean, we only discovered this in the early 90s, and it really, really heavily influences the stress response, as well as the immune system and a whole lot of other systems in the body. It actually, what they found is kind of underlies a lot of systems and influences other areas of biology that we just didn't know had these kind of underlying factors associating them, or influencing them. So FAAH breaks down this particular endocannabinoid, and anandamide activates the CB1 receptor, which we'll talk a little bit about later on here. And just as a kind of a cool note, when they discovered anandamide, they named it after the Sanskrit word for bliss, because when they gave rats high amounts of it, it mimicked the effects of them being totally blissed out, which makes a ton of sense when we kind of look at these different variants here. So, and from this chart, you can see we've got anandamide. FAAH comes and breaks it down into arachidonic acid, and arachidonic acid is actually a polyunsaturated fat. So this gene also influences your fat metabolism. So we'll see that in these variants as well. So what are the two variants here? The C allele has higher levels of FAAH, and subsequently they have lower levels of anandamide because it gets broken down by the higher levels of this enzyme. And vice versa, the A allele, they have lower levels and less functional FAAH enzyme, and so they have higher levels of anandamide naturally. Now, let's talk about the higher levels of anandamide first. These people from the studies show that they're generally less anxious. They have a better stress response. There's a nice study that shows that they have a better response to PTSD extinction therapy, where you're trying to kind of avoid or try to disassociate the emotional charge from a memory. So these people are able to sort of recover from traumatic experiences a little bit better. But at the same time, people with this particular variant, it's associated with higher rates of anorexia as well as non-cannabis drug use. But in terms of cannabis, these people tend to use cannabis a lot less, less than half of the other variant, about 11% versus 26% for the CC variant, from a 2,100 person study, which is a pretty big population to look at. That's pretty good data. So they have less preference for cannabis use in general, and they also have a better response to a low-fat diet, interestingly enough. Now, the CC allele, who has less anandamide and more FAAH, they are strongly associated with craving cannabis, craving pot. Greater craving after abstinence. Brain scans, which I'll show you in about 2 slides, show higher activation in the dopamine reward centers of the brain when exposed to the smell of cannabis smoke. And I am a CC, and the first time I smelled it, it just lit up my brain, and really had that very low level biological response to it. And we'll start to see why that might be as we add more genes and start to see how these things work together. And they report feeling happier than AA after cannabis use. And it's even stronger when combined with these cannabis receptor SNPs. So they also show, interestingly enough, worse sleep with cannabis use, but only if they have symptoms of depression. It's a pretty kind of fascinating three-way kind of variable thing there. And they also have a better response to a high monounsaturated fat diet. So that's things like olive oil, avocados, peanut oil. This is from a study that was looking at this particular gene variant. And you can see here that this, I love how this is just labeled happiness in the study, but they were giving these people a subjective reporting kind of happiness scale before they smoked, and then at regular intervals within 60 minutes of them smoking. And you can see that in general, the A allele, which has more anandamide, more of your natural endocannabinoid, was reporting higher levels, statistically significant higher levels of happiness overall, just baseline, compared to the C allele, which has lower levels of that cannabinoid. And you can see that after they smoked, within 5 minutes, the C allele levels out to the same level of subjective reported happiness as this other allele type. And that sustains throughout the entire session. And as the A alleles continued to stay high for the duration of that session, they reported a lower subjective happiness level, which is, really, really interesting. If you know people, or you're one of those people that doesn't have a good response to cannabis, and you just don't like the way it makes you feel, you might be an FAAH variant. And these studies really have opened my eyes in terms of how differently people respond to it, just because it's, when you see data like this, it kind of cements things. But let's look at another chart from the same study. So this is also looking at these two alleles and withdrawal and craving symptom severity. So before they stopped smoking, I think this was like a 7-day follow-up. So both of these groups were smoking. They were about the same level. And after they stopped smoking, the FAAH group, or A, that also already has naturally higher levels of cannabinoids to begin with, they couldn't care less. They actually even looks like slightly lower, though it's not significant. But they didn't really have much withdrawal symptoms or anything. But on the other hand, the C's reported about 1.5 times more, 1.75 more times more more craving and more withdrawal symptoms. So, that's really significant in terms of, thinking about what it's like to stop smoking. And, this might provide some strategies for some people if you're trying to cut down on cannabis use, as well as understanding just the general endocannabinoid system and what this means for things like stress response and anxiety. Now, the conclusion here is the influence of cannabis on our endocannabinoid homeostasis really impacts our desire to consume cannabis. So, you already have this balance in your body, and depending on where you're at in that balance, that's really going to influence the desire to consume cannabis and intake, THC and other cannabinoids. So, CNR1, and we're going to kind of circle back to FAH here, because there's a great study that really looked at both of them together. And so this codes for the cannabinoid receptor 1. This is the main receptor in your brain and central nervous system that gets activated when you ingest THC, and as well as some other cannabinoids. And it's a little bit complicated. The science, it's still being done to really understand how other cannabinoids impact these things, because up until now, they've really only looked at THC. There's not even any genetic studies that I could find on CBD. which was kind of surprising, but it just leaves so much open to further discovery. And so there's several SNPs that have been identified within this gene that have really significant impacts on people's response to cannabis, as well as a whole lot of other things. This is a highly researched gene, and one that's just continuing to be kind of compounded on with all of these different things. It's involved in anxiety, feeding behavior, how full you feel. So in terms of the munchies, this gene is pretty impactful when it gets activated versus when it's inactive or less active. It also is involved in neurotransmitter balance, and they've shown that blocking this gene increases anxiety. PTSD patients show lower levels of your natural endocannabinoids, anandamide and 2-AG, AE, that should say AE. And they show less CNR1 activation in general. So the cannabinoid system is depressed in PTSD patients, which is one of the reasons why cannabis has been shown to really help some of these people manage their symptoms. And what's important to recognize here as we're talking about this is this perspective, this genetic perspective, really can help us understand why maybe it doesn't work for everyone, but it really should help hammer home the fact that it does work for a lot of people, and there's biological reasons why. So people that are involved in the legalization side of it, I think incorporating some of this information in would be very helpful in strengthening your argument, and sort of cutting through some of the dogma that's been passed down over the years about this. So activating these CB1 receptors decreases intestinal permeability, which, if you're kind of follow the health world, it's been a pretty big topic the past 5 or so years, the concept of leaky gut, because it impacts a lot of different systems in the body. But actually activating these CB1 receptors can really help manage that and offset it. And that's something to pay attention to as well. It's also been implicated in cognitive function, bone density, constipation, weight loss, circadian rhythm, hormone regulation, and etc. I mean, this gene really has a lot of impact. So let's look at a specific SNP here. We're going to talk about 2 of them that they've identified. So this is RS2023239, and this is probably the main CNR1 SNP, but there's a few others. And the G allele here is associated again with more craving and withdrawal. And the effect is stronger when it's combined with FAAH allele, particularly the CC. The G allele is also associated with greater CB1 receptor density, which we'll kind of talk about in a few slides here. So this is from a study looking at the effects of both CNR1 and FAAH together on the brain response to marijuana cues. So they were exposing people to marijuana smoke or showing them a joint and scanning their brains to see what the response was. And they were specifically looking at these dopamine reward centers of the brain. And what they found here is that the people that had more of the CNR1G allele or the FAHC allele showed higher activations, almost double the amount of activation in these reward centers. And you can see here, there's someone with an outlier that shows 3 times the amount of activation with 3 of these different alleles together. So when you're combining 2 genes, there's 4 alleles to look at, and they didn't include what would happen if you had 4 alleles, but I think you can kind of extrapolate that this would continue to go up. And so that's pretty powerful. I mean, when you think about why people seek cannabis, this is really starting to get to these underlying biological roots, and why. So here's my kind of working theory on this: is that if you have more CB1 receptors, but you have less anandamide to activate those receptors, the combination of that is going to lead to a lot more craving and seeking out a substance to balance that. It just makes sense. And, again, from a legalization perspective, if we take this kind of biological approach, it starts to really lessen all of the sort of the moral stuff that gets thrown around here. It's some people, it can be very helpful in terms of, really balancing these systems in the body. And the other thing here is that, as more of these genetic studies come out, I think we're going to see a lot more strain development and extract development that is specifically suited to different people's genes. And I'll talk a little bit more about where I see that going. But that's something that I think is kind of the next frontier here. So again, this is the same gene. This is from a different study. Now, the G allele is also associated with lower hippocampal volume among heavy cannabis users. So people that smoke a lot were shown with this particular allele to have significantly lower volume in this area of their brain. And this is an area of the brain involved with memory. And this is a chart from that study. And this is actually a study that was done on cadavers. They took their brains out and weighed them and figured out the size of these different areas, or the area of the hippocampus. And so you can see here, these are both controls, people that were not smoking cannabis in their lifetime. And interestingly, the people with the G allele actually showed higher hippocampal volume than people with the A allele if they weren't smoking cannabis. And this is statistically significant here. Now, on the flip side, people that smoked cannabis that had the AA, they showed not much different. This isn't statistically significant between this column and this column, but you can see there's a pretty big difference here between this column and this column. So, this kind of gets into the area of, understanding the potential risks if you have a certain variant and really weighing the pros and cons. Now, when I say pros and cons, if you're using cannabis to treat a medical condition, and, there's really a good reason to consider, sacrificing some of your memory, then, that's the decision you and, your healthcare practitioner should make. But having this information is pretty telling. But again, you really can kind of choose to look at it in two ways. One is that these G alleles come with a greater risk, or you can look at it as the AAs, that's protective of memory loss and lower brain volume. So, I tend to look at it on the positive side of this myself. If you're an AA, good job. But at the same time, it appears that the AAs just have lesser preference for cannabis in general. So, it's a pretty interesting factor to take into account. Okay, now this is a different SNP in the same gene. This is RS1406977. G carriers show they have lower amounts of the CNR1 RNA, meaning they just produce less CB1 receptors. And they also show worse working memory than AA. And here's a chart. You can just see the difference in that RNA expression, which it's a pretty big difference here. Now, this was a study that was done looking at working memory. So that's short-term memory. And they're administering a test called the two-back test, which, participants were shown a series of images or shapes, that kind of thing. And they had to remember what they saw 2 shapes ago. And you can imagine as you start to compound that, it gets pretty difficult. Now, the initial reportings for people that the control group that didn't use cannabis, the difference between the AA and the G is almost non-existent. It's not statistically significant, pretty much the same. We're talking about 2 points difference in test. So that's basically the same. Now, people that used cannabis, and I believe this was acute, like right after they smoked, the AA subjects compared to the G subjects, their memory, their ability to recall things in their short-term memory didn't didn't change at all, but the G carriers showed a really significant drop. And you're talking, 20, 30 points here, 25 points about. So that's something that's really significant. And we're going to talk a little bit more about memory here because there's a lot of studies that have been done on that. And also just to clarify, these studies are looking at cognitive performance and deficits, because that's something that's been, again, easier to get grants to study. And, I think we're going to see, what I really hope to see is studies that begin to look at the medicinal benefits of cannabis relative to people's genotypes. And I think that's really where we're going to see, kind of a sea change in the way people think about this. And, I'm really hoping that this information that I'm presenting is going to help kind of move that forward in some small way. So, with this particular SNP in the CNR1 gene, G carriers show worse short-term memory with cannabis consumption, and the AA is protective against these negative memory effects. Okay, so the next gene we're going to talk about is something called ATK1. This is a protein that acts as a signaling molecule in the dopamine pathway. So it's not a dopamine receptor, but it kind of influences the way dopamine is received by those receptors and moved along in that pathway. The C allele of this particular SNP, or 2494732, is associated with more quote unquote psychotomimetic effects, meaning effects that mimic psychosis. And, it's kind of a gray area with the medicalized language around this, but you can interpret that to mean that these people experience cannabis as subjectively more psychedelic. They might experience things like time dilation or visuals and hallucinations, and it's a little more paranoia inducing. So you're kind of looking at the overall intensity of altered state experience. And the C allele shows much higher self-reported symptoms, quote unquote, related to that. So you can see here the TT genotype on this inventory they gave these people after smoking. And actually, this was an excellently designed study. What they did is they let people bring their own cannabis to the study, and they even analyzed all of the strains that these people brought for THC to CBD content, so they could isolate that as a variable and kind of check it against all these other things to see if that influenced this response. But what they found is that the only thing that they could isolate was this AKT1 genotype. So you see with the people who are carrying the C allele, they reported, maybe about 15-20% higher, more kind of psychedelic, altered state, intense kind of effects here, with the CT reporting a little bit higher. And sometimes that happens. It's not always, a linear effect. It sometimes can be a little bit unpredictable with that kind of response. But overall, the TTs don't experience cannabis as intensely in terms of those more psychedelic effects. Now, this gene also interacts with the dopamine receptor gene, DRD2. And this particular interaction is really, really interesting. And there's a lot of research, and there's been a lot of research around cannabis related to schizophrenia, because it's been known for a really long time in the medical community that people with schizophrenia tend to be pot users. I mean, I think it's like 70% of people with schizophrenia have a history of cannabis use, as opposed to like 20% of the population. And there's a lot of discussion around whether that's correlative or causative. Perhaps these people are trying to balance something out again in their endocannabinoid system and are seeking cannabis as a way to do that. But this particular gene is really, really interesting in the way that it affects the development of something, really important to keep in mind, especially if you're a practitioner working with medical cannabis. But the CC allele here of the ATK1 gene uh, just in general, people have a 2 times likelihood of being diagnosed with a psychotic disorder, but only with a history of cannabis use. And the daily cannabis use bumps that up to 7 times risk. And it interacts with this dopamine receptor to continually amplify that risk. And here's a chart looking at that from a study. And, this is something in general that while it's kind of uncomfortable for people who are pro cannabis to talk about, because it's a negative aspect, I actually think it's kind of, there's an onus of responsibility to recognize that there are some risks that come with cannabis use for certain people, and I think the for certain people part is really important. And we can use these genes to really assess people's tendencies and potential for problems with cannabis use. So you can see here they're looking at a combination of the DRD2 gene and the ATK1, and the more risk alleles you have with just general lifetime use, this goes up. The potential to develop psychotic diagnoses goes up 7 times. Whereas if you have these 2 quote unquote risk alleles and you don't use cannabis, you actually almost have a 0% chance here. So, if you're a practitioner and you're working with, say, a PTSD patient, and, they have a history of trauma and other co-occurring mental, problems, it would be really wise to look at these genes if you're thinking about prescribing cannabis for them, because, if they have 2 of these risk alleles, that's probably not really something you want to risk. And I think it's just kind of a responsibility for people that are really looking to make this substance useful for people. And again, here we see this go up with daily use. I mean, this is a massive jump here. So that's a pretty powerful study here. COMT, this gene is a really well-studied gene. It's a really cool gene. It has a lot of implications outside of cannabis, and we'll talk about that here. But the COMT, it contains instructions for making something called catechol-O-methyltransferase, and it's an enzyme that breaks down dopamine and epinephrine and other similarly structured compounds in the body, and it regulates dopamine levels. Outside the brain, it's also involved in detoxification and hormone regulation. And this particular SNP, the 4680, is kind of nicknamed the warrior versus warrior allele. And we'll go to, we'll come back to why that is. But the GG allele here produces the most COMT, and they have the lowest levels of dopamine. The AA produces about 3 or 4 times less COMT than GG, and they produce higher levels of dopamine. And AG is like right in the middle. And this gene is something that's stratified really nicely in the population. About 50% of people are AGs, and then 25% of people are GGs and 25% of AAs, just to give you an idea of kind of how that breaks down. But this is kind of a summary of the other studies that are out there on the COMT gene, where you see low dopamine levels are associated with impulsive behavior, especially high risk behavior, anger, addiction, and food cravings. And then on the other end of things, dopamine excess is associated with schizophrenia, anxiety, insomnia, paranoia, chronic pain. So you kind of see here that it's not really good to have too much or too little of dopamine, and you really want to kind of be in the middle here. But in relation to cannabis, and so, in general, this is also something related to the ATK1 and DRD2 genes. If you have someone who's a COMT AA, you probably also want to take that into account. But as far as cannabis goes, the COMT GG variant showed a 40% reduction in working memory after consuming cannabis as compared to the AA. And the AA showed only about 11% lower than baseline after consuming cannabis. And also the GG showed impaired episodic memory, which is talking about long-term event recall, things like your personal experiences, whereas the AA variant showed no impaired episodic memory compared to not being high. And here's a chart from that study. And you can see at baseline, there's no significant differences. It's a little lower, but it's not considered technically significant by the statistical analysis. Now, after they gave people THC, I believe this was another oral THC study, which, you know, if really, I'd love to see another study looking at smoked cannabis, whole plant cannabis, that kind of thing. Because there are some differences. If you're familiar with something, it's called the entourage effect. It's looking at the interaction of all these other endocannabinoids in cannabis, and there's been over, I think, about 130, a little more than 130 individual cannabinoids that have been identified, and you know, there's really only good research on a couple of them. So just to give you an idea of the potential for where this type of thing could go, and isolating you know, what the effects are of these particular cannabinoids. And there's some good research that's being done, but there's just not any yet looking at these specific types of genetic differences, because these studies tend to get done once there's already a basis, you know, in the literature. The genetics get added in after the fact. So as we see, you know, more of a relaxed research climate, we're going to see more of these studies. But anyway, so after these people took THC, you can see this is about a 40% decrease in their test results. And this is something called the digit span backwards test. This is essentially people having to remember a string of numbers and then remember the digits backwards. And if, you know, you can imagine how hard that might be if you're high, if you're a GG variant, and, you know, if you're someone who can smoke cannabis and you don't experience any of those memory deficits, you might be an AA. The AA here showed about an 11% reduction, but still very, very little. You know, so that's pretty interesting, and it really really gives us a picture of how differently cannabis can affect people cognitively. You know, I would love to see a lot more studies in this vein with, you know, different strains and that kind of thing, but and those things are coming. But this is really interesting. So, you know, if you're someone that loses your keys after smoking all the time, you might be a GG. So that kind of concludes the genes that I'm going to go in depth with. I don't want to take too much of your time here, but just to give you kind of a summary of other known cannabis-related genes right now. The COX-2 gene, it's another gene that has to do with working memory in cannabis. It also interacts with CNR1. CYP3A4 is another liver enzyme, and it metabolizes CBD, although there aren't any SNP studies out there. So if anyone knows of any, please send them my way, but I haven't been able to find any, and hopefully those are coming down the road here. SLC66A4, that's a serotonin transporter gene. And it has to do with decision-making skills with cannabis consumption. So they were administering something called the Iowa gambling test to people. That's kind of a standardized decision-making test. And they found that the different variants of this had different results on that test. ABCB1 and PENK, these are both strongly associated with chronic cannabis use, probably more so than any other genes, especially the PENK, in terms of just people's overall lifetime cannabis use. And MGLL, this also has an association with chronic cannabis use and likelihood of developing trauma after PTSD. And MGLL is very similar to the FAAH gene, except that it breaks down your body's other main endocannabinoid, 2AE. 2AE is the name of that endocannabinoid. And so you can see that this is influencing people's innate endocannabinoid system and that response and resiliency to stress. And MAPK14 influences the white matter volume in the brains of cannabis users. So this is similar to what we looked at with the hippocampal volume, except we're looking at white matter. So the two different variants have, one has a reduction in white matter and the other one doesn't. So those are what's out there right now. And I would say this is far, far, far from comprehensive in terms of what will be done. But, it's still really exciting research in terms of understanding this individual response here. So conclusions here, I'd say it's safe to say after looking at this that the human response to cannabis is highly varied and at least partially genetic in origin. Studies estimate that cannabis use is between 30 and 70% genetic, depending on what study you read and how they're interpreting that. But there's definitely a significant genetic variable here. In my opinion, it's really time to have a more informed discussion about cannabis, taking this individual variability into account. And, it really adds another dimension that, allows us to look at it from more of a scientific lens, again, without this dogma attached to it. In terms of saturated fat, we looked at, 25% of the population, they're 60% more likely to get Alzheimer's and 7 times percent more likely beyond that if, they eat high saturated fat diets. So should we ban saturated fat? No, that'd be insane. So, I think that just because something has a really negative effect on a particular group of people, we should be talking about that particular group of people versus the other group that maybe it has a more positive benefit for. This research is really just beginning. Like I said, there really haven't been any genetically stratified research studies on the medical benefits of cannabis. And when you look at things like cancer, autoimmune disease, PTSD, seizures, and neuropathy, and all these things that people report having positive effects with cannabis, you have to also take into account that for some people who've tried it, it hasn't helped them. So if we're able to understand what the genetic basis for these differences and why these things work for certain people, we can really customize people's treatment protocols and really utilize, cannabis as a medicine in the same way that, you might prescribe one drug for one person because they respond a certain way, and you might respond to a different drug to a different person. And, this idea and approach is really still pretty new in mainstream medicine. I mean, we're starting to see more testing, more genetic testing related to drugs. SSRIs with psychiatry they're starting to test for those because there's big differences in the way people respond to them. I would really love to see this information get incorporated into the cannabis industry as well. I think it's only going to help everyone, strengthen the argument for using it as a medicine for the right people at the right time. And additional research, once we start looking at other constituents in cannabis, like terpenes, the things that give it flavor and taste, which have been shown to have really, really potent effects on the body, just on their own, as well as the over 130 other cannabinoids that are in cannabis. How did they affect people, and then how did they affect people on a genetically stratified basis too? That's really exciting, because then we get into the area of breeding strains that are going to have different cannabinoids, higher levels of certain ones that you might, from a genetic test, be able to tell, well, this one is better for this person versus that one. And what's the dream? I think it's a genetic analysis that matches you with the perfect strain here. I'm really excited to see where this research goes personally. And I hope this helps kind of, give you a different perspective and hopefully a more informed one. So that kind of concludes things here. And, this is the point in the webinar where the internet marketing gurus tell me I'm supposed to pitch you my services, but out of respect for your time, I'm not really going to do that. What I will say is that if anyone wants to have a conversation with me, send me an email. Happy to talk about anything related to this presentation, as well as, if you're interested in looking more deeply into your genes, whether you're working with a particular health issue or just looking to optimize your overall health. My area of expertise is kind of cognitive performance and really, developing lifestyle and dietary habits that reinforce that. So if anyone's interested, feel free to send me an email and we'll talk. I do free consultations and, we'll figure out if there's something I can do for you. But that kind of concludes the webinar here. Does anyone have feel free to type any questions. I'm going to go back through these comments and see if there's any ones that I can answer in particular. Mark says, with everything you said about being AAC, AC, or CC, why do people say they get higher as they get older? Is it just a tolerance issue? Mark, that's a really good question, and it's something that I haven't found a very clear answer for. I imagine that there is a strong epigenetic component to that. I do know that with people who use cannabis regularly, the CB1 receptors tend to have a dulled response. So perhaps that might play into it. I think there's a lot of factors there. I don't have a clear answer for you on that, but I think we're going to see some clearer pictures being created around that as more of these studies get done and really look at the epigenetic factors involved here. So let's see. ApoE4, does this affect MS patients using CBD? I'm honestly not qualified to answer that question. I really don't know, Mark. But actually that's something I'm going to look into. I'll look into the ApoE4 if there's anything out there on interaction with CBD. That's a really good question, actually. Casey says, where would I look on 23andMe to find Wish I Am? You can go to, when you're logged into your account, you can go to the top toolbar and click on tools and then raw data, and there'll be a search bar that you can search for different RS numbers. So all those RS numbers that I put into this presentation, you can copy and paste those, put them into that search bar, and it'll tell you whether you're an AA, AC, or CC. Okay, well, it doesn't look like there's any more questions, but I really appreciate all of the comments. Thank you, Yuri, and everyone else, Yoav, Brant. Good to see you. Really appreciate you guys coming and checking this out. This research really, this information really means a lot to me. I'm very passionate about it. So I'm really happy that you guys sat through this, and if this is something that you guys found valuable, I'm going to send out a little survey after this, and I'd really love your feedback on what I did well, what other information you might like to know, and if this is something that you'd like to do in the future again. So thank you so much for coming. I'll be emailing everyone here a replay of the webinar so you can watch it again, share it with your friends, and yeah, thank you guys. I'll see you later and hopefully talk to you some soon.